This is the first page. Hello, everybody. This is class number 6. If I understand correctly, we have this is the 4th week, no? And if I understand correctly, we have an exam at the 6th week. I am correct? Am I correct?
Yes, yes, you are correct.
Okay, so good. I am focusing very much on the on the okay, let's say practical side. Okay, you will discuss how practical they are, but certainly I am not talking about the deep theory, at least not before the midterm. I am skipping some theory that if we have time at the end of the course, we will teach. But right now, I am focusing just in the practical side. Okay, I mean, the minimum practical side. So, if you don't have any question before starting, I think I give you a homework, no? The one homework was reading a paper that I hope will be useful for today. And yeah, I will probably give you another homework like reading papers or watching videos or something like that. What we did in the last class, in summary, we defined some words about looking in database. We defined the keyword query and subject, which are words that come again and again in bioinformatics. I mean, in this kind of I mean, people working in databases have different names, but in bioinformatics, these are the names used. In general, people look about searching patterns in text, but in bioinformatics, we say query in subject, which is equivalent. Then we say that searching database it's easy if the sequences are identical, like you are looking in a dictionary and you essentially look for the same word. If they are identical, fine. But if they are not identical, if you are working in a biological context when you have mutations or change and evolution, then we are happy with partial match. And we define the idea of looking for things that are not identical but are similar. So I told you about two tools that people use, Hamming distance and Levenstein distance. Levenstein distance is also called edit distance, in case someone asks you, which is good to know. It's not necessarily something that you're going to use because these are not the distance that are the best for bioinformatics, but they give you the background. Okay, Hamming distance essentially count mismatch. Sometimes you use it. Okay, it may come back again in another problem. Then Levenstein distance count
Sorry,
mismatch and idle. Yeah.
Atakan text me to join the class.
And do you have the invitation? No, shall I send it? Can you send it?
He wants to link.
Yeah. Do you have it?
I don't know where
Yeah,
to
me
put
neither.
it.
Let me see. There is a button here. There is a button like invite. In participant, there is a button called invite, and I can copy invitation. And let's do something. Let me send you in the chat because I don't want to stop very much. Yeah, Atakan. I always forget that there is people that is not in the course.
Trial version.
What is it? Trial version. What trial version? Oh, not good. Okay, so let me send you this. This is the code. If you can make it, maybe you can send it to him. No.
Okay, I'll send it.
Fine. Thank you very much. I guess that you. Well, it depends. Sometimes only the host can invite people. I don't know. Okay. Thank you anyway for telling me. So we were doing the summary of last class. There is another word here that I should have emphasized, indel. Indel means, question, what do you mean indel? What do you mean by indel?
Insertions or deletions.
Insertions and deletion. And why we write that way? Why we don't say insertions or deletion? The basic point is that in this context, we don't know what is an insertion, what is a deletion, because essentially you have two sequences and they are both contemporary, and one has something extra that the other doesn't have. And normally there is no any way to know that if that sequence is an insertion in one of the sequence or is a deletion in the other one. So insertions and deletion for us are essentially the same thing. Okay.
What do you mean by contemporary?
That they are today, that the sequence that you have today, you have Escherichia coli and Bacillus subtilis, they are existing today, and they have, let's say, similar genes, except that in one bacteria, the gene is longer, has an insertion, and in the other has a deletion. So insertion and deletions are actions that normally happen through time. And since we don't know what happened in the past, we don't know if that sequence appeared because of an insertion event or because of a deletion event. You follow me?
Okay, yeah.
So, contemporary means in our type. Well, in the sometimes contemporary means that both are in the same time, they share the same time, but not necessarily our type. The point is that we don't know which comes first. We cannot say that Bacillus subtilis gene is evolved from the E. coli gene or the other way around. We don't have that relationship. We don't know. I mean, at least not with the tool that we have here. Maybe looking with the genome, with the archaeological data, maybe you can find something. Maybe you can find, if you find like a time machine. Dot plots. This is a key idea that, in fact, we don't use it very much in the practice, but we use to understand what is happening. I mean, it can be used in practice. Sometimes people use when you want to finally understand what is comparing two sequences or something like that. Sometimes it's useful. But for us, the most important thing is that this is what is happening behind. I am essentially trying to explain you how the tools work inside without necessarily doing everything from the tool. And then there were two concepts that we discussed, global and semi-global alignment. Do you remember the difference? Say something. Global and semi-global, yeah?
A different length of sequences in a semi-global alignment.
Yeah, well, different, very different, let's say, because in general, in all these alignment, they will have different lengths. Sometimes they have the same length, but in general, they have different lengths. Now, the different length can be in the semi-global is typically you have a gene and you are comparing with a genome, something that is kind of several order of magnitude bigger. And then the second point, what is the modification that we did in the alignment in that case? So, or let's say, what is global alignment? This is the exam question, you realize, no? And in fact, it's kind of one of the important things that, in fact, it's kind of the point of these two classes, what the difference between global and semi-global and local alignment. This is a super point. So anyone else can try to say what is the idea of global alignment?
I think at global alignment, we try to match two long sequences match.
Okay.
But at local alignment, we just try to match short sequences.
Okay, you are partially right. Welcome, by the way. You are partially right because it doesn't correspond exactly to the length of the sequence, but to the length of the alignment. In the global alignment, you try to align the two sequences together in the total length. So that is what Atakan calls long. So global means you can put gaps anywhere to make them compatible, and you stretch in the idea of comparing from the beginning to the end. Then in semi-global, since one of the sequences is shorter, the query is shorter, the main difference is, what is the main difference? Okay, they will deliver a packet.
Trial version.
Why did I say trial version? Did we run out of the money for the university? Are you hearing that they say trial version?
Yes.
Yeah.
That is very funny. Okay, maybe we didn't pay, maybe we ran out of money. What I was saying that there will be a delivery. They got a message, so you will excuse me if they ring the bell and deliver me something, okay? So what I was saying is you have, in the semi-global alignment, we distinguish internal gaps from external gaps, okay? That is a keyword. We have gaps inside the sequence and gap outside the sequence, and outside the sequence don't count, okay? So internal gap have a price, external gaps are free, okay? Have a cost if you like. Fine, move your head, say yes.
Yes.
Okay, I am afraid that if we are in the trial mode, they will turn off in 40 minutes. So I don't know what we'll do in that case, but we will find a way. Everybody's in the forum, no? So I can send you an email if we need to connect again, or I can send you some. I don't know what happened when I write you in Axis. Do you get a text message, or do you have to connect to Axis to read the things in Axis?
The latter.
Ah, yeah, that is why I never get anything from there. Okay, so back to the class. So we show Levenstein distance, also edit distance. And in general, distance, we say that distance is essentially means that small numbers are better. Closer distance, they are more similar. We say essentially if two things are similar, they have distance 0. And the more different they are, the more chain they have, they are more distant. Okay, so that is the philosophy of looking for distance. So, but then when, and well, we also say this point about alignment that we just discussed, different alignment for different question. I have two definitions: global alignment, compare gene to gene or protein to protein. Well, you read it, no? Semi-global and local. We discussed this. And I have this other definition that I have in another slide. All sequence must match. All gaps are penalized. Penalized, you know, like penalty, like the penalty in football, is a punishment, or in other words, a cost. The penalization here means that there is something bad happening. Okay. In the semi-global, external gap are not penalized, and in local, that Atakan described, some part of the sequence must match some part of the other. You are not looking for matching everywhere. You are looking to match something. External gaps are also free, and there are two kinds of local alignment, with or without internal gaps. Initially, people did without internal gaps, and it's kind of the one that I'm going to explain today. So there is this plot that I found in Wikipedia that tell the idea of global versus local alignment. In the global alignment, as you see, you want to match everything to everything. If some part may be longer, some part may be shorter, but essentially you want to match equal to equal. In a local alignment, you want to match one part of the query with one part of the subject. And as you see, you may have two matchings inside the same sequence, okay? So that is important. Given two
Is it
sequences,
necessary for global alignment to be at the exact same length?
no, because you have gaps. I mean, what I mean is this: the sequence don't need to have the same length, but after putting the gaps, they are the same length. Essentially, the alignment works putting the relevant part of the same length. How you make them the same length? You have essentially two things that you can do. One is you put gaps to make the shorter sequence longer, or you trim the borders. for example, in semi-global alignment, we trim the borders that are not important. We forget what is outside the alignment. We only care the part that is in front of the query. In the local alignment here, they don't need to have the same length, but this part that are aligned will be similar, and after gaps will be the same length. Okay, after gaps they have the same length. And this part we don't care, we ignore. This part we don't care, we ignore. And this other part they have to match with gaps. Okay. So the short answer is they don't need to have the same length, but they tend to be similar length. Okay. Now the point to keep here is that you may have more than one alignment in or partial alignment, more than one, what is the name? High scoring pair, that is the technical name. Here, you have an alignment here and an alignment here. And these are two alignments in the same query and subject, okay? That can happen. Now, you can say, for example, if you want, you can include this green part and say this is mismatch and have a long one. But somehow this is going to be good, this is going to be bad, this is going to be good. And if you don't take the bad part, the good part will be better. Kind of the alignment in this side and this side are better if you don't take in consideration the part in the middle. See, this may be proteins and the conserved part are domains, or this may be genomes and the conserved part are genes. Okay? Yes, no? Yes?
Trial
So,
version.
Part of the query must match part of the subject. I found, well, I made this example yesterday. We can make another eventually. So how you match? Here you may realize that there are two ways to align. I showed you one, but we can do another that I want to show you in a minute. But the point that I want to show you is this. First, external gaps don't count. So I didn't put anything here, and I didn't put anything here. The alignment, here I am representing with these 4 lines. Okay? You see them?
Yes.
So part of the first sequence matching part of the second, and the rest, we don't care. We ignore in this part. Okay? Now, the point that happened here is this. We, since the external gap do not count, if you are trying to make the shortest distance, you will always find something like this. I can match A with A and have one perfect, and this is perfect. A matching with A, and the rest we don't care, so the distance is 0. Okay? So what people found, and it took a while for people to realize this, is that since the smallest, I mean, the point is that distance doesn't work for local alignment. People know the idea of local alignment, but they could not solve it thinking with distance. You see? You see why? Because distance is a minimization problem. So we are trying to find the minimal number, the smallest number. And in the case of local alignment, there is always the solution of I don't take anything, nothing to nothing that has distance 0. Or you just find one matching letter, or even if nothing matches, you can still say, okay, they have distance 0 because I put gaps everywhere. And the gaps outside don't count. So we need a different approach. And this is one of the ideas for today. Instead of calculating a distance, people say we are going to calculate a score. And the difference is that a score is something that you want to make bigger. Instead of having a small distance, you want a big score. So instead of having 1s and 0, like we had before, counting 1 with mismatch and 0 with match, So we will have now a positive score for when the two letters are equal, a negative score when you have letters that don't match or you have a gap. Okay, so the score increase when things are good and the score decrease when things are bad. Okay, the philosophy is the same, but with big numbers instead of small numbers. So that is why we now say that we are looking not for the distance, but for the optimal alignment. Optimal in the sense that we are going to find the minimal distance. Sorry, not the minimal distance, the maximal score. That is what I say. Instead of looking for the closest one, we are looking for the optimal alignment, the maximal score. Oh, I'm saying it back. Okay. In principle, this is not a big change. Instead of looking for a small number, we look for big numbers, kind of changing the sign of the problem. But in practice, it works much better because now we want to have big scores. So you essentially you want to make your alignments longer, because the longer you can make them, putting positive number, the longer the score will be. Okay. When you calculate distance, you are trying to take things out. You are trying to get rid of things. When we are doing the maximal score, you're trying to put things in. Everybody should come because we want to have a lot of score. Okay. Move the head.
Okay.
Okay. Yeah. Make some gestures that I know that you are listening. What else? So, if you, I didn't make the plot here, unfortunately, but we can, I have something to show you anyway. So instead of placing ones or zeros, now we will put we will put positive and negative numbers. Let me see. I have something here in the, I have an Excel. Maybe I have this also in the in Google. In Google, but let's look at this one. I don't know if the colors are good. Maybe I didn't choose the best colors. They look funny now. Do you see this? I will make a zoom first just to show you the idea, but then I will zoom out intentionally. If the colors are not very good, let me try. Okay, one question. Is the color decent, good or not? Can you see? Because I mean, I forget that some people are blind, color blind, and they don't see the difference between red and green. Yeah, Ahmed is laughing. What? Yes or no?
It's okay, I think.
I can see.
Okay.
Okay.
Let's see if I can change to another color range. Color, let's say I will go from blue to yellow, green. Maybe this will be better. Okay. Okay. Yeah. Well, anyway. Now, you want to see the big picture. You want to see the kind of landscape. So here I have a parameter that I can change, the 1 for match and -1 for mismatch. So the old case was mismatch was 1 and match was 0. That is what we have the dot plot that we discussed last week, no? But now we are changing just a little bit the values. But what do we win here? The trick is now, remember, we are essentially looking for diagonals. So you start here and you move in the diagonal and you go adding. So 1 plus 1 plus 1 plus 1 plus 1 plus 1 plus 1. So what you will find is that at the end of the line, most of the numbers will be the numbers grow. Okay, before going to the next slide, I want to show you the other idea here. What is the other idea? Now we put positive and negative number. What is the other idea? Is this? once you get this dot plot or dot matrix, if you like, a dot plot matrix, I will call it, because now it's not only black and white, it's with different number, we can find the optimal score. And then this is the idea that you have to keep in mind. In the global alignment, we go from corner to corner. In semi-global alignment, we go from side to side. And in local alignment, we got any diagonal. Okay? So if we were doing a global alignment, you have to start here in the first corner. Here the sequence are going left to right and top to bottom. So this is the first. And you essentially try to find which is the way that you can. This is kind of a video game, no? And you have to jump either horizontally, vertically, or diagonal. You cannot go back, and you go jumping, and you try to collect. So if you go to a negative number, you lose point. If you go to a positive number, you win points. So you try to jump and get to the other to the end and see what is the score that you get. There are many ways that you imagine this is a city, and you have to move in different ways. So some corners, some streets are cheap, some streets are expensive, and you try to, or let's say you find money in some streets and you pay money in other streets. Okay, and you want to choose the path that will give you more money at the end. You see the philosophy here? Okay. Some people are looking like, I don't understand anything. I don't know if they are a video.
So if we move diagonal, that means we're doing local alignments, no?
No. The difference between local and global is where you start and where you end.
Can you show an example, quick example? Because it's...
Yeah, I just say this is a global alignment. In global alignment, you start from one corner and you try to get to the other corner. Okay?
Okay.
Now, there are many ways to do. You can go all horizontal and then all vertical. You can go all vertical and then all horizontal. Or you can jump in the middle, try to jump horizontally, vertically until you get to the diagonal. Then you jump to another and you get to the other diagonal. Then you move, I don't know, until here, you go here. Imagine this can be easily a game that you start here and you have 3 buttons, horizontal, vertical, diagonal, and you have to jump. And you can have a competition who get from one part to the other with the maximum number of points.
Okay.
You imagine that game?
Last
I
week
can
we
imagine.
started from the bottom left, but right now you are starting.
Yeah, that the people complain. Why people complain? Because in the drawing that I showed you last week, this sequence was written bottom to top. And the reason for that is very silly. Matrices are written with the numbers growing down. But plots are drawn with number growing up. It's just a convention. When you write a matrix, this is 1, 1, this is 2, 1, 3, 1. This is the row numbers, row 1, row 2, row 3, row 4. Okay? When you write a matrix, a data frame or something like that. When you do a plot, usually the Y number grows when you go up. But the philosophy is the same. In the last, essentially, you have to remember where you started the sequence. If you started the sequence in the bottom going up or in the top going down. But it's just a visual thing. Point is, you in the global alignment, you start in one corner and you want to get to the other corner, trying to collect the maximum number of points. Okay? Okay, homework, make a video game. Now, second alternative in semi-global. How we do in semi-global? As we say, in semi-global, we go from, what happened? 1 second, one second. In semi-global, we go from one, what I did? I changed the screen. Here. In semi-global, we find the largest sum from side to side, from, in this case, top to bottom side. Essentially, because moving horizontally here is free, because the moving horizontally here will be the external gap. So you get, for example, here, and then you jump, or here, and then you start jumping to this diagonal, and then eventually you want to get from anywhere. I mean, you can choose the starting point freely here. Then you jump vertically, horizontally, diagonal until you get to the other side. Okay? That is semi-global. In global, the starting point is fixed, the ending point is fixed. In semi-global, the starting point is free in the first row and the ending point is free in the last row. Okay? Now, what Ahmed said, in local, you are even more free. You can start from anywhere and end anywhere. Okay? So I will choose, for example, to start here, and I will move through this diagonal and end here, and I will collect the points. Okay?
Okay, now I understand why it's called local. It's
Exactly.
inside the, okay.
It's inside, exactly. It's local. It's not global. Global is meaning essentially you have to take everything from one corner to the other corner. Local, from anywhere to anywhere. And semi-global is something in between. You start somewhere in the first row and you end somewhere in the last row. Okay? Now, I don't want to enter too much because it's not very relevant right now, but the point is that you can essentially make a formula that takes the value of each point plus the up, down, and you can get something like this. If you see here, the diagonal, you get the number here. Like, this is essentially assuming the value that you have plus plus the neighbor. And the maximum value I got is here 18. What I'm doing in this second, what is the name? The second spreadsheet is I am taking this value and adding them diagonally. Okay. And when I do that, I can find essentially what is the cell with the highest point. That will be the score of the best local alignment. Okay.
Is your rule to add 1 from diagonal to diagonal when you go from diagonal to diagonal?
No, I am essentially adding the value that you have here. In some cases, the value is 1 when they are when the okay. Let's go here. When the number in the column and the letter in the column and the letter in the row are the same, you add 1 because it's good and you get good score. When they are different, you take -1 or -2. It depends on the formula. You know, you have some freedom, and this will be a subject later. But I can say that mismatch is not -1, is -4, for example. So it's very expensive to make a mistake. So you can play with these names, how much you win when you are correct, how much you lose when you are wrong. That is the kind of thing that people like to play. Oh, yeah, it's too small. And then the number will change. Okay. But right now, the bottom line is that local alignment is about starting anywhere and finishing anywhere and collecting points. You want to make a score, a number that is going to be big. Okay. The score will have a positive value when things match and negative value when they mismatch, and then even more negative value when you have gaps. Okay? Question?
I'm sorry, but in the second sheet, I didn't get the green parts.
In this second sheet, what I have, the color, this is what is here? It's called the conditional format. It's an Excel trick. And essentially, I say that go from red to green. Red is minimum. The color is depending on the values. The minimum is going to be red, and the maximum is going to be green. It's a trick to make it visually easy to find. If you don't like that color, let's go from, we say blue to yellow to green. So the lowest value will be blue, the middle will be yellow, and the high will be green. Maybe you can find a nicer green like this one. This is all Excel magic. So the blue cells are very bad, and the more green they are, the better they are. In this case, this is the biggest one. Okay.
It's the value of the score, right?
That is the value of the score.
Okay.
Yes. And then you have to go back and find what is the alignment. But you know that the alignment is something that ends with this one. And you kind of get an idea how to go back. Okay? In fact, I think I have it later. Okay, we will discuss this point later. Tamam?
I don't understand how we jump from positive to positive when we go diagonal.
Okay. Let's say that, think this again like a video game, and you can jump in 3 directions. If you are in a given cell, let's make it a little bigger. If you are in a given cell, you have 3 buttons. You go down, you go right, or you go diagonal. Okay? So if you are here, I can go down, right, or diagonal. If I go right, I lose 4 points. If I go down, I lose 4 points. So I go diagonal, and I get 1 point. So let's say I start here. I get 1 point. Good. What is my best move now? Here. I get 2 points. What is my best move now? Well, I can go here. I can go here.
Well, if you add all these values, you can just make your path longer. You can go this way and then down.
Yeah,
we got a certain starting point here, no?
In the local alignment, you are free to choose a starting point, and you are free. But you realize that if I start here and I go collecting points, I will get some point. But if I start before, I get more points. Your goal is to get the maximum point. Now, Ahmed has a point, and it's the part that I am not showing you here. The point is this. If I can move diagonally, horizontally or vertically, I can make like some tricks here. I can make some ways to get more points. I can like get 1, 2, 3, and then more points here. Yeah, you are right. But that is not what we want to do. So how we solve that? Well, you should remember something that maybe is not obvious. What happens when you move the horizontally to the right?
Basically, you don't.
Gap.
Yes, gaps.
Yeah, you are putting a gap. Therefore, there is another rule that says essentially that moving horizontally has a price. You pay points for moving horizontally. Moving vertically is kind of free. Sorry, moving diagonally is kind of free, but moving horizontally and vertically, you pay extra for the fact that you are getting a gap. So, for example, let's say if I start in the first letter, let's start here. I will have a C and a C. Okay, fine. Then if I move diagonally, I will have a in the first, so I will have CA, and I will have CC in the other. So I have here, when I am here, I have one mismatch. Then let's say I move another diagonal. It will keep moving diagonal until here. So I will have CA GT, and the other is CC GT. So as long as I move diagonally, I am getting face to face. I am growing in both sides. I'm growing in the horizontal and in the vertical. When I move diagonal, I advance in both. Now let's say I am here, and now I want to go down here. So I will have a T in the second one. Here, I am here, no? Can you see the screen? No? I am here. So I move diagonally, and then I move vertically. That means that I win this T, but what I put in the horizon, in the first sequence, I have to put a gap there. Now I am here, and I can go with this diagonal. I go, let's say, I have T with T that I had before, then the gap, and now I have A with A and C with C. So this is A and C, and the second is A and C. So if I start in the first corner and I get until here, this is the alignment that I'm making. Okay? And I calculate the score. Let's think this way. If I give you this alignment, if I show you this alignment, this is a way that you calculate the score. Okay? If I give you C, A, G, T, gap, AC, you can start here. You get 1 point here, -4, +1, +1. You pay the price of the gap, and then +1. So we need a gap cost.
Sorry, why we don't see the gap?
Because I'm showing you the simple model. The formula gets much more complicated, and I don't want to enter in that formula right now. The point is that there is another, you have to consider three things. Basically, the score that you get when they match, the price that you get when they mismatch, and the price that you pay when you have a gap. Gaps typically are more expensive than mismatch. Okay? Any question?
How do you interpret the gap cost to the formula in score sheet? I mean, how does it know when to put a gap and when to not?
Let's say that there is a formula that I don't want to explain today because I don't want to spend too much time in this technical thing. I promise that I will show you later. But the basic point is that there is a formula that allows you to fill all this matrix. Essentially, you have to make a decision. In every cell, you have to decide what is the best I can do. Shall I come from the top, paying the price of the gap? Shall I come from the left, paying the price of the gap? Or shall I come from the diagonal, paying the price of mismatch?
Okay.
Those are the three alternatives. In every cell, or there is a way to explain this. You are in the last one. How you get the score here? Well, you had three possibilities. You either come vertically, horizontally, or diagonal. What is the best that you can do? Okay, you compare all the possibilities. And then you realize that to calculate the score of each one, you have to go back. Essentially, you realize that to calculate the best option to get to the last corner, you have to calculate the three neighbors. And then to calculate the three neighbors, you have to calculate the 9 neighbors. And you realize that to calculate the last number, you essentially have to calculate everything. Okay. So the point that I want to make is this. First, the result was this matrix when you have the rows of the query and the columns of the subject. Okay. Second, what you put inside the matrix depends on a scoring schema, a scoring rule that give positive value to match and negative values to mismatch and gaps. Okay. The details of that formula we are going to discuss later. I don't want to discuss now because I don't want to distract from the goal of the practical things. Right now, what you need to know is that the tool that we are going to use has these rules inside. Okay, that essentially what all these tools are doing is when they do global is corner to corner, semi-global is from top to bottom, and local from anywhere to anywhere, mostly in diagonal. Okay, why mostly in diagonal? Because diagonals are free, and horizontal and vertical they have gaps. Okay. Yes, no?
Yes.
Yes.
Okay, okay. Say yes, say no, say something. No, this one, this one. Okay. Find the largest sum in any diagonal. Yeah, the simple way to think about local alignment that you move only in diagonal. That is called ungapped local alignment. Okay, if you cannot move horizontally or vertically, and you can only move diagonal, then the best thing that you can do is you start in a point and you can only move diagonally and you get the score here. But that means that you don't have gaps. Local alignment without gaps is easy to understand because you only move in diagonals, okay? And you essentially try to find the best diagonal. With gaps, it's a little more complicated, and in fact, people don't know it very well. Okay? Why we care? What is the tool that we are going to use? And you probably know this name. Yeah. Now, what is the point here? There are 2 points here. One is BLAST is the tool that we are going to use today. And the other point is BLAST is local alignment. This is the message here. People tend to use BLAST thinking that BLAST is global alignment, and it's not. BLAST is local alignment. Okay. So it is super important that you get used to the idea that local alignment are a partial match and not a global match. Okay. Now, the first version of BLAST, in fact, had no gaps, only looking in the diagonal, and that is what we say here. You start here, and you can only move in the diagonal. No, I don't have a diagonal bottom, but you can only move from here to here, then here to here, and try to find the maximum number. Okay. The second version, they put gaps, but it's much more complicated. Tamam?
Tamam.
That means the following. You will have BLAST results even in sequences that are very different. The only thing that they need to have is something in common. For example, I don't know. Let's say that humans and chimpanzee or gorilla are very similar. Okay? We are very similar, and we are kind of globally similar. But humans and, I don't know, octopus or fish have eyes, and maybe human eyes and fish eyes are similar. So BLAST will find that. BLAST will find that humans and fish have similar eyes, or similar bones, or something like that. But it's not a global thing. Just one part, like, I don't know. The tail of the rat is look like a human finger, or the BLAST, for example, will tell you that the trunk of an elephant is similar to a human hand. because they can use it to hold things. Okay, because they are locally similar. They don't need to be similar in the complete context. So you have to take care with the result. The result that will tell you doesn't say that the two things are identical. They say that they have something in common, but not that they are the same or even they are similar. They have part of them that is similar. Tamam? Okay, I have a question here. In fact, I will show you something, but I will ask a question in because it will help me to explain the next slides. Scoring matrix. As you say, as we discussed, there is a parameter here that we have to choose it how much you get when you have success and how much you lose when things are different. How much you get when you are in a match and how much you lose when you are in a mismatch or a gap. So these things are typically written in a matrix. So here is my question. I can explain this theory with this slide that I'm writing like in R. We have two letters, X and Y. X and Y are variables here. And if they are equal, the score increases by a positive number. If they are different, the score decreases by a positive number. If one is a gap, the score is reduced by 2. Okay? Tamam? So then you can make a matrix X and Y that represent these rules. Okay, the matrix will have like a ACGT in the rows, ACGT in the columns, and in the center, in the cell, will have 1 or -1 or -2. Okay? You get the idea that these rules here can be written in a matrix. Now, my question is this. I can write this slide with this notation, or I can write like in mathematics. You see, these two slides say the same thing, except that in one is written in computing language and the other is written in mathematical language. Which one is easier for you to understand?
I think the first one.
For this slide, I think mathematical is a little bit clear.
I don't know. They tend to be shorter. And if you are used to it, it's more clear. But if you are not used to it, I mean, a lot of people kind of panic. They say like a mathematical letter, then say, they have a panic immediately. So since we don't want you to have a shock, like a epileptic seizure, like, mathematics. I don't know. It's dependent on you. I really think that it's important for everybody to be comfortable with mathematics. I'm not saying that you should necessarily enjoy it, but you should be comfortable. Like, I mean, like any physical activity, you should be comfortable moving, even though you are not a professional marathon runner. You should have a very basic thing that, okay, I can go to the corner and I can carry the bags from the grocery, no? Okay. Maybe you can write in the chat telling me if you want, let's say, R syntax or mathematics syntax, okay? By the way, write your student number in the chat so I can carry the attendance. Okay? In fact, the last, you can write everything in an equation like this. And you didn't vote anyway. You tell me later, or well, if you write like in R, in math, or in a single equation. When you get used to, this is the most clear because it's clean of every noise. But you need practice to see this.
Isn't it either X or Y is minus a gap?
No, X or Y are letters. Are, for example, ACGT.
Yeah, I know, but
So
when equation is equal to -2, you said x is a gap or y is a gap.
yeah, if you are aligning a letter to a gap, you get a score of -2. If you are aligning two letters, if you are aligning, yeah, you are aligning A to A, you get +1. A to C, you get -1. Gap to C, gap means that X is gap, then you get -2. A to gap, you get -2. What I'm saying is here, if one of the letters is a gap, the score is -2.
Yeah, but when we actually get -2 in the equation with the sequences, how will we know where is the gap?
Ah, okay. That
Is
is
it
a
on X or Y?
Okay, okay. I see your point. I see your point. The point is here. Here I am talking about the what is the name? The substitution method. I'm not speaking about the Excel thing here. I'm not speaking about this. I'm speaking about this. Okay. So in this model, horizontal gaps and vertical gaps, or let's say gaps in the query and gaps in the subject, have the same penalty. So we don't distinguish in the term of the cost. Now your question, if I understand correctly, is this: if I knew that the optimal score is here, how do I reconstruct the alignment? Well, I have a slide later, I think, but the basic point is that calculating the score is one question, and calculating showing the alignment is a different question. So let me say it again. When you are looking, when you are comparing two sequences, you have essentially the freedom to put gaps. And when you put gaps, you will get some score. The trick is that finding the best score is straightforward with filling this matrix. And after you fill the matrix, you have to do something called trace back when you fill and complete the gaps. And that is kind of harder. You need to do it after you do the first thing. And what I will show you later is that it can even have more than one solution. You may have several alignments that have the same score. In other words, the best score can be done in several ways. So we have to be tricky. You have to be careful to find an alignment that is biologically meaningful. The system will give you different results that they have the same score, and some of them will make more sense biologically than others, and you have to be careful with that. Okay.
So basically, we can know there is a gap or gaps, but finding the place of the gap is another story.
Exactly. Okay. You find the gap. That we will do. But in many cases, you don't care. In many cases, you just care what is the score. You don't care exactly what are the alignments. Let's see. What I mean is that there's a good question that we will solve soon. Okay. Okay, let's finish this so we can have a break. So the scoring matrix, that also called substitution matrix, essentially one row and one column for each letter in our alphabet. Alphabet here means the sequence of, not the sequence, the set of letters that we are considering. For us, alphabet in DNA is the ACGT. That is the DNA alphabet. Okay. In amino acid, you have 20 letters. Okay. If you want, you can expand it with one extra letter, that gap. Okay. But essentially, alphabet means the letters that are valid in this context. So for example, the typical thing that people has is 1 for match, -2 for mismatch in DNA, -1, -3, 1, -1, or even 4, -5. We will discuss them later, but let me show you this. This is, for example, something, one of the standard things that we use. This is the scoring matrix for DNA, ACGT, and you have positive when they are equal and negative when they are different. These matrices are typically symmetrical because, I mean, replacing A for a T is the same as replacing a T by an A, essentially because they don't have any order in life, no? They don't have any, what I mean. Substitution is something that happened through evolution, and again, we don't know how evolution happened. For amino acid, you have bigger matrix, of course. So, and in this case, the frequencies are, I mean, what I mean, the scorings are different, are based on the frequencies found in nature. This is a discussion that we are going to have in more detail, but let's start with the historical thing. The first person who did this thing, they made it by hand, was Margaret Dayhoff in 1978. And she invented the idea of point mutation that are accepted. Accepted here means accepted through evolution. So what she did is this. At that time, there were not many protein sequences, but she collected kind of all the proteins that were known, and she grouped them in things that were very closely related, like hemoglobin in different organisms. Okay, and in general, you can have any mutation. A mutation can happen, but most of mutations are lethal, meaning that the organism doesn't survive. Okay, if a mutation is accepted, then you will see it in the nature. You start with one sequence, you change letter randomly, most of the organisms die. The one that doesn't die are accepted. Okay, accepted by evolution. So she found sequences that were kind of 85% identical, and they say, okay, if they are very similar, whatever mismatch they have are a mutation that essentially is, this is the assumption that this mutation happen in a single event. The basic problem here is that if you see, let's say, leucine and phenylalanine, you see two proteins, you are comparing two proteins, then they are identical except in one amino acid. And in one case you have leucine, and in the other you have phenylalanine, for example. Phenylalanine. Well, anyway. So what is the story there? What they have assumed is that if the sequences are very, very similar, that single mutation happen in one step. You went from leucine to phenylalanine without any intermediate step. In more distant sequences, in sequences in proteins that are evolutionary very separated, you may have intermediate step, like leucine went to methionine or to isoleucine, and then it went to phenylalanine in a second event. Okay, so you say, okay, I will make a single mutation event. So she defined this, and this is one of the clever ideas. She created a matrix called the point accepted mutation 1 with the rate of substitution for a short period of time. What is the chance that that amino acid change in a very short period of time? And then with mathematics, with a very easy mathematics that we're going to discuss after the midterm, she calculated the what happened at 2 times, 3 times, 30 times, 250. So it is not very clear what is a short period, but just to fix idea, this is not clear because evolution times can be different, but let's say that in 1,000 years, you have one amino acid changing. Let's to fix idea, okay? So what she calculated was what are the chances of changing from every amino acid to another amino acid in a short period of time, 1,000 years, let's say, okay? Then, if you know what happened in 1,000 years, you can simulate 2,000 years. Essentially, you go from today to 1,000 years, and then from 1,000 to another 1,000 years, and then they got the second matrix. And then you do it again. Essentially, they are multiplying matrices, you know. So you get the matrix of 1 year, then the matrix of 1,000 years, 2,000 years, 3,000 years, 30,000 years, 250,000 years. So the bigger the PAM number, it means that you are assuming that the sequences are more distantly related. Small PAMs mean that the sequences are closely related. Big PAMs mean that the sequences are distantly related. Okay. Now, the matrix look like this, and I wanted to show you this. And they found something that is super interesting. And first, what we see here. Tell me what you see.
I see some zeros.
Okay. What else you see? I'm always surprised that people see things and they don't see. What do you see?
So
You see a square matrix.
If two similar amino acids match, then the score is higher. If they're far away from each other, the score is
yeah, that is the super result. Exactly. That is super result. But there are simple things that we have to say. First,
and why is there a star at the end? What's
okay, you see a star. Let's say there is a star at the end. The star here represents the gap, if I am not wrong.
Is it the rule to prepare mattress, or
we
didn't have any map in the in your Excel sheet?
No, but okay, just be careful. This matrix is not the Excel sheet matrix. You have to, that is a typical confusion. This is called the substitution matrix, score substitution matrix. This is not the dot plot matrix. Okay, these are different. How they are connected? Let's see. If in Excel, here I have a nucleotide, but if I had amino acids and I have I don't know, a T and a C, what is the score that they will put here? In DNA is very easy because you have two possibilities. They are equal or they are different. There is nothing else. But in protein, you have a story that is much more complicated. In proteins, if you have a T in front of an A, you can have -1. But if you have a T in front of a C, you will have -8. And you have a T in front of a, I don't have G here, so these are all the options. If I have an A in front of a C,
There's
I have -6.
G.
There's a G?
Is this coming
okay,
from
yeah. Okay.
The way they are acidic amino acids or basic amino acids?
Yeah. That is a super good question. So, okay, again, first thing that we see here, let's say this, we see a matrix. We see a matrix that is square. The names of the rows and the names of the column are the same. Okay. So when you get an alignment, you will get a letter in one sequence and a letter in the others, and one letter in the query, let's say this one, and a letter on the subject that you will go in the column, and you will find the cell that intersect the letter in the query with the letter in the subject. And the number that you find here will be the score of that pairing. of that pairs of letters. Okay. In other words, that will be the letter that we put here. When we build the dot plot matrix, the number that we put there depends on what of the letter in the column name and the letter in the row name. Okay. For DNA, the rule is simple. Positive when they match, negative when they mismatch. Okay. For proteins, the rule is more complicated. It depends on which amino acid. Then you find something. When the two letters are equal, in other words, when we are in the diagonal of this matrix, mean that the letter in the query and the letter in the subject are the same, then you get a positive number. In general, a positive with a positive is good. I mean, conserve the amino acid is good. Okay. Then, as Ramal said, if they are similar, typically leucine and isoleucine, in here they are not so identical. Anyway, but in some cases you have like this one, Q and A. I don't know what is Q and A, but when they are conserved, Q with Q is 8, A with A is 8. But if you replace a Q with an A, you still get a positive score. Okay. Now, Ahmed say this is the basic or acid. Well, and yes and no. Yes, it is true that the numbers that are positive, like here, N and D, the amino acids that have positive score, they share chemical properties like hydrophobicity and, well, essentially the chemical properties. They share chemical properties. But the interesting thing is that this matrix doesn't take any chemistry in the model. When they make the matrix, they didn't think about chemistry. They only look at the frequency of sequences. Remember, Dayhoff took sequences found in nature, and she counted how many times leucine changed to isoleucine, on how many times. And based on the frequencies, with some mathematical rule, she found these numbers. And what you found
these numbers
that the
are
number
constant, no? They don't change. I mean, let's say 2 isoleucine to methionine, it's 8, let's say. It's a rule?
Yeah. Given the matrix, when you choose a matrix and you get a rule, the point is that there are many matrices that you can use. And the matrix will depend on the problem that you want to solve. Okay?
I don't understand the logic of the numbers. Why is it like -6 when P meets G? Why is it like 0 in some places? There is no negative or positive. I don't
Okay. The complete answer, this is the integer part of the logarithm of the probability of substitution. But that is a longer story. We will talk about that, but we need to define some probabilities, and I don't want to do that before the midterm exam. So
Is it about codons?
No, this is amino acid to amino acid. No, we don't see codon here. Right now, what we need to know is that for amino acid, you have several options of the scoring matrix. And the scoring matrix essentially tells you how much you win or lose when you pair an A with an I or an A with an S. That number are in general negative because changing an amino acid by another is in general bad, but sometimes they are positive or neutral. For example, if you replace T by S or S by T, you don't win, you don't lose. Okay? According to this matrix. And this matrix is built based on the observed frequency in a particular set of proteins, the protein that were available in 1978. And with some hypotheses, you're saying, okay, I try to make it in a very controlled way. I take the best example that I can do by hand, and I count the substitution in the simple case, and then I extend with a mathematical rule to the future. Now, there are people, and with good reason, say these are not necessarily the best ideas, because essentially what Dayhoff was doing global alignment of proteins, and therefore you can only see proteins that are very, very similar. So in 1992, there was there was a couple, if I'm not wrong, Henkoff. I'm not sure if father and son or husband and wife, because I don't know if Georgia is male or female. Anyway, they got a different idea. Instead of doing global alignment, they'll do local alignment, and they did the same mathematical thing, but only with blocks that are conserved. That is called block substitution matrix. So again, Dayhoff took proteins and she did global alignment. To do the global alignment, you need to have proteins that are very similar. So she separated all the sequence in 71 groups or 77 groups. So she did 77 group of local alignment, of global alignment, and she found the frequencies in all these cases, essentially in a manual work. At that time, most of the thing was doing by hand. And in fact, the local alignment was also found in 78. Like the paper I give you, the Smith and Waterman paper, is from 1978. So she had no option of doing local alignment because local alignment was not yet found. In 1992, people knew better. They say, okay, we are going to do local alignment. And now, instead of comparing protein to protein, we are comparing domains to domains, if you like, blocks to blocks. Therefore, the idea that the domains can evolve at different speed. So here, since the speeds are different, you cannot take them directly to like in PAM that you have evolutionary distance. So essentially they say, okay, I am going to classify my matrices based on how similar the groups are. And the typical one that people use is this one. This is the default matrix that we use for protein alignment. It's called the Blossom 62. That is based on alignment that has 62% of similarity. The numbers that you see are different, but they have the same philosophy. Again, a square matrix, positive and negative numbers, also integer numbers. We have to say these are integer numbers. X mean that we don't know. X is not an amino acid, it's a convention that we write when we don't know. And this is, if I understand correctly, is the gap. And what else? Well, you see the same thing that happened. Now, isoleucine or leucine, they have positive score. So most of the numbers are negative. The numbers in the diagonal are positive because keeping the amino acid is good. And changing an amino acid by one that is chemically similar will give you not so good, but still positive. So keeping leucine by leucine is the best. Changing leucine to isoleucine is not so bad, but changing to other things is bad. Well, okay, maybe valine. Maybe this J, I don't know what is. Okay. So you see the story in proteins is much complicated. You have much, what does it say? Much thing to keep in mind. Okay. Matrices. Let me talk about gaps. I should have a slide here. Have gaps. Let's speak about gaps. First point is that you want, from the biological point of view, you want to say that there is a substitution here instead of saying, look, there was an insertion here and a deletion here, and there was an insertion here and a deletion here. Kind of the story that you need to explain this alignment is much more complicated than saying, okay, this is just a T changing to a C or an A changing to a G. So in general, we want to use substitution before and gaps only as a last resource. Okay, if you can align things using only substitution, you do it. You say, okay, the letter change, and you don't put gaps a priori. So in general, gap is greater, and I think not in general, always. There are other reasons, but the basic point is that you want the gap to be greater than mismatch. Okay, so it should be always be worse to put a gap than to have a mismatch. Then, and this is a little more biological. We have these two sequences, and here I have 3 A's, and I am matching this A with the first one. But I could also, if all gaps have the same value, I can put a gap here and put the A in between. Same here, I have a G. I can put this G at the first or at the second, or I have many ways to put, and I can kind of put them inside. Okay? Notice that here, in the way that we define the gaps, these two alignments have the same score. Yes? You have the same number of matches and the same number of gaps. We don't have mismatch here. And the number of gaps is the same in both cases. Do you agree?
Yes.
But biologically, the first alignment makes more sense than the second.
I have a problem again.
What is your problem?
These two are different sequences, not the different showing of the same sequence. These are totally different.
What do you mean with this? What do you mean this? The first and the second, or the
The one, yeah,
one,
and
two,
the yeah. These are different.
what?
I mean, we say we prefer this, but we say it like it's the
Okay,
same
in the first-
thing,
in
and
the-
we
in the-
prefer the first one, but it's not the same thing.
Okay, I don't know what you call the thing, because I have many things here. So let me
With the
put
gaps
names.
and the insertions, deletions. For example, when you say TCA, gap, GA, this is a different sequence, and the one in the bottom, TC gap, A gap, this is a different sequence. These are different genes or genomes or something.
Okay, you have a point here that is a language problem. The sequence don't have gaps. The alignment have gaps, and this is an important difference. The sequence is TCAAA GAG GATA. TCAAA GAGGATA. The sequence are the same here. The first sequence in every block is the same.
Okay.
The last sequence in every block. The alignments are different because the alignment puts the gap. The sequence are something physical. The sequence, you go to nature and you capture, you got the sequence. It's something that you can hold with your hand. Okay? In theory, you hold with your pipette. The alignment is something that exists, let's say, only in the computer, because we have to guess where to put the gaps. Okay? It's an important difference that it causes a lot of confusion, and it is a super valid question because it happens a lot. So the alignments are different, but the sequences are the same. Moreover, both alignments have the same number of gaps. I have 3 gaps and 2 gaps, 5 gaps here and 5 gaps here. Okay? Do you agree? Last point. What do you think is more logically from the biological point of view?
Maybe mutations can happen like locally, not separately all over the place, but locally, someplace there's a mutation and it comprises 3 letters, let's say.
Yeah.
Rather than 3 letters in the beginning middle and the end.
We have to be careful with the word mutation here, because we have two kinds of mutations. We have substitution and indel, or let's call insertion. But you are in the correct direction. We have a mutation event, an insertion event, or a deletion event, if you like. Maybe deletion is easier to think. And the way that people think is this: if you start, I mean, how this thing happen? You have the polymerase that is copying, and for some reason, it may be the polymerase jumps, and there's something that you don't copy, and then it binds again, and you keep going. That is how you get a deletion, like the polymerase is jumping, or you miss some nucleotides in the copy before closing the gap, something like that. But there is some kind of mechanical disattachment of either the replicase or the what is the one that is kind of the DNA polymerase, or something happening during the replication process. So here the story is like, here we have one event, and here we have another event. We have two jumping polymerases. Here we have 5 jumping events. Here you jump 1, and then you jump again, and then you jump again, and then you jump, and then you jump. So from the biological story, this alignment kind of doesn't make a lot of sense, because you are essentially assuming that the polymerase is jumping all time. Here, you minimize that hypothesis. You say the polymerase jumped once here and once here. Okay? Now, in other words, we want few long gaps instead of many short gaps. And that is something that we have not discussed before. Okay? I'm not going to enter too much in detail, but this is a point that you have to keep in mind, that from the biological point of view, you want long gaps. Now, how people do that, the keyword that you'll find is called affine gaps. Affine, well, this is the explanation that I give you. Once an indel event starts, it can grow. But if the polymerase jumps, then it can, I mean, if you open a gap, it is hard to open a gap. But if you open a gap, it can grow easily. So you essentially separate the gap in 2 parts. One is gap opening, and the second is gap extension. Okay? Having a gap is expensive, but once you decided to have a gap, having a long gap is cheap. Okay? The basic idea is that you have 2 values here. Without entering too much detail, so you have 2 values: the initial gap penalty, the price that you pay for opening a gap, and the extension penalty. So, and this formula is called affine. If you have a formula like the cost depend on the length of the gap, is called linear. If you have a fixed cost and then an extension penalty, that is called an affine gap. So right now, I just want to give you this keyword, linear and affine. Affine means that you pay a price for opening the gap. And that was, no, that we can stop here. Okay, this is the question that was asked about how to put the gaps. That is called traceback. And people were asking this question. So the summary of what we say is this. Given the problem, you will choose one or other way to define a substitution matrix, like to give you the score when you put one letter in front of another. For example, you put A in front of T in nucleotides, you will have some rule. If you put A in front of T in protein, you will have another rule. That rule is codified in a matrix, like the PAM matrix or the Blossom matrices, or the DNA matrix. Okay. Then you will have also to decide what is the gaps, how much you punish gaps, how much you punish opening a gap, how much you punish extending a gap. Given all that, you will have a way to calculate you will fill the matrix, not you, the program will fill this dot plot matrix with the score corresponding to the substitution matrix, and they will find a path, a trajectory inside that dot plot matrix, and you will get a point and you get the best score. In particular, when we do local alignment, we go from any starting point to any ending point, trying to find the maximum total. Problem is, when you get the maximum score, you may have more than one path from the starting to the ending point that give you the same score. In other words, there may be more than one solution. So if you only want to know the score, that is true in many cases, you don't spend time doing the traceback, doing the position in the gaps, for two reasons. One, because it's expensive, and second, because it's some ambiguity. In very few cases, you want to see exactly the gaps. In many cases, you just want to know which is the closest sequence. I mean, for example, if you are comparing your query with all the database, you want to find the score, and only for the one that got the best score, you do the traceback, not for all of them. Otherwise, you spend a lot of time doing traceback of sequences that you don't care. So the philosophy of the traceback is more or less like this. This is a global alignment. By the way, Needleman-Wunsch is the paper that defined this algorithm. You start building the matrix from the first, building the alignment from the first corner until the last. And this is the score. In this case, the score is 0 for this alignment. And then you want to go back. So essentially, you need to know what was the decision that bring you here. What is the best thing that happened before? Here you have A with U, so they don't match. So you have a mismatch, -1. Therefore, the previous one was 1. And if you got 0 here and you have a mismatch, it means that the previous score was 1. So you are here. Here you have C with C, that is good. And then you have to know where you came before. You either come from here, or from diagonal. Now, if you move horizontal or vertical, you have a gap. So you are paying a penalty. So the only solution is that you came here, diagonal. And then essentially you have to record, the program has to record which was the arrows that they used to move in every step, or with some other rule, like trying to uncalculate the gap, the score, and find how you get. But here, if you see, there is two solutions. You can either move this way or this way, and then this way. So when you have these ambiguities, that you are putting gaps in different places, you will have different solutions. For example, here, as you see, this gap can be put, what is this gap? You have a G with G, then this horizontal is this gap. So here we have A with A. In this cell here, we have an A with an A here. Then you have what do you have? Either T with T and T with G. This one, A, T, G. If you go this way, you go A, T. By the way, I'm not hearing anything. Can someone talk to verify that my audio is working?
Hi.
Okay, good. I was not sure if I was speaking alone. Okay, so if you go by this way, you get this solution. If you go here, you have a gap. The first one, you have a gap in the first, and then you go T with T. And well, you see, the summary of this is that the same score can have more than one solution of gaps. And in this context, we don't have any way to decide which of these alignment is the, let's say, the correct one. The correct in the sense of the biologically meaningful. At some level, they are all equal. We don't have any way to distinguish them. So they are all true at the same level. Okay?
We go from top left to bottom right so far, but arrows showing the other way.
Because you have two parts. We go from top left to bottom right to get the score. And after you got the score, you trace back. That is why we call trace back to put the gaps. It's something that you do after. It's done in two steps. First, as you say, from top to bottom to calculate the score. And after you did, you go back. And the point is, when you go back, you may have more than one path, more than one way to do it with the same score, with the same value. Okay? And in this approach, we cannot distinguish. We don't know where is the gap. In other words, the gap is something that we put in the alignment. The sequence don't have gaps, we put the gaps, and in this case, we don't know which is the, I mean, they are all equal for us. The difference, if you want to put a difference, the difference here is kind of what is the biological story that you tell, what is the evolutionary story. So you need something else that is not with this information. We need something else to choose between these. And that is what we call biology. Okay, that was the traceback story that was pending. The other point that I want to make is that the alignment score depends on the substitution matrix. Okay, I mean, it should be obvious, but if you have Pam and Blossom and they have different names, or if you can put that the mismatch of DNA is -1 or -4, the value will depend on the matrix. If you change the matrix, you will have different scores. So one question is, which is the best matrix to choose? We will talk about that later, but the point that I want to make is that different matrix will have different scores, and eventually different score can even give you different alignments. Okay, in other words, if you change the scoring matrix, you may have a different alignment. The bottom line I want to say is this. Ideally, that is not always true, but ideally, when you publish your alignment, you should say what were the matrix that you use, how you choose what was the scoring matrix that you use, what were the price of gaps, the price of mismatch. Otherwise, I cannot make the same alignment, or I can choose a different matrix and get different points.
So can we say if we want a strict alignment, more precise alignment, we make the penalty higher than
yes,
very higher than the other one?
Yes, exactly, and that essentially the question is why you want to do that, why you want to do a strict alignment. And normally the reason is because you are working in a context when you know that the two sequences that you are aligning are close heteroevolutionary or something like that. You want them to be very close because you expect a small number of mutations. So every mutation should be very expensive. I mean, if you expect very little number of change, very little number of mutation, or even more, every little number of gaps, they have to be very expensive. In the other hand, if you expect the sequence to be much more distant, for example, you are taking sequence from bacteria that are found in many different places, or you are comparing human to Saccharomyces, for example, that you can do that. You know that they have some genes in common, but they are so distant that, in fact, there is some mismatch. You will not be so worried if there is some mismatch or some gaps. So the strictness, as you say, is connected to the score, but the reason to be strict is about what is your hypothesis about the evolutionary distance between the sequences. That is kind of the reason that we have to keep in mind when choosing the matrix. Or in some cases, people do the alignment with different matrices and see what is making more sense at the result. Now, After you fix the matrix, the question is, what is the best score? Because if the sequences are identical, well, everybody's happy. But if the sequences are not identical, they will have a high score. But how high is high enough? Is kind of a question that was asked last week. What if they are very different? What do you mean very different? Or what is the threshold to say that two things are similar? So in general, you need to make some kind of hypothesis to answer that question. The most common hypothesis is a statistical hypothesis. I mean, in some cases you have another, like when looking for primers or something, you can make other hypotheses. But in general, when you are looking in a database, you make an hypothesis that essentially that the larger the score, the less hits you will find, the less successful, I mean, the less subjects in the database will have that score.
But if we find that, it means it's very much we want, right? It's the...
Well, not necessarily, because let's say that I got Martian DNA, let's say, but something different. Or well, Martian, there is a more classical example. Let's say that all your database is made in Europe and you get DNA from Australia. This is a very good case to keep in mind that I always use, because Australia was isolated from Europe for a long time, and therefore they evolved. I mean, you see, even in big animals, you have kangaroo, you have koala, you have many animals that exist only in Australia and nowhere else, and many animals that exist in Europe, Africa, they don't exist in Australia. So when you want to compare them, let's say you got kangaroo, and you want to compare with all the animals that you know in Europe. Some animal will be closer. I mean, probably, I don't know, dogs will be closer than rats, let's say, or than elephants. You know, dogs, you have rat, you have fish, you have mouse. Of all these animals, what is the closer to kangaroo? Dog. Does it mean that kangaroos are dogs? No, it's just the closest one. Now, how close they have to be? Well, as is again the word that you use about how strict you want to be. If you are very strict, then you have to be I mean, if you are very strict, you will have very few sequences that match. For example, let's say that if you accept, let's put it this way. When you get a kangaroo, you find that it's not identical to anything that you saw before, but they share some attribute with some animals. So at some level, you will say, okay, this is a mammal. It has warm blood, and they have fur, and they don't put eggs. This is a mammal, so you will find it similar to all mammals, more or less. But if you are more lax or less strict, you will find that it's a vertebrate. They have bones, and they have a skeleton, and at some level they are even similar to fish. Okay. And if you go more and more relaxed, you will find that it's, I don't know, they have a central symmetry. They are animals. They are not plants. They are not yeast. They are very distant. So the more lax you are, the more hits you have, the more similar thing. In the other side, the more strict you are, the more condition that you put, the less hit you will have, the less result you will have. Okay? So basically, maybe what you want to think is like, I want to have a score that is so high that I will have very few results, because I believe that this result will be meaningful. Okay. Now, idea number one, large scores give less hits. The more higher your score, give you less hits, less results. Then, this is the interesting part. We can make an estimation. We can make a guess, an educated guess, a mathematical guess of what is the number of results that you will get in a database, assuming that the database is random. What I mean by random here, this of course will be discussed in more detail later, but you may have the chance that you find two sequences that are similar just by accident. For example, if you are not careful, you will find that bats look very similar to birds. They fly, they have wings, they have behavior that is similar. So if you are not careful, they will say, oh, they have the same attributes. But in a second perspective, bats and birds are not related. They just happen to have something in common just by accident. So the statistical hypothesis is this. If I have a database with letters that are chosen randomly, you just put letters A, C, T, G randomly, let's say, what are the chances that I will find alignment with this score? So if my score is if with a very small score, I will have a lot of hits, a lot of results that are just random. If the score is higher, I will have less and less probability of having a hit with that higher score. Okay, so the big hypothesis here is that you are comparing your alignment, assuming that the database is not biological, it's just random. Okay, that is a big idea of statistics. Maybe one day we will speak longer, but the basic idea of a statistic is you find something and you want to say, is this something that I expect to see I mean, just by chance, or it is something more meaningful? It is something that I don't expect to see it by chance. It's like if you want, you open the window and you see at the clouds, and the clouds have some shape. And you see, oh, look, there is a cloud with a particular shape. So you may say, okay, there is something happening here that the clouds have a particular shape, or you can also say, it's just you happen to find some pattern in the clouds. Or if you see the star, the constellation, and when people see bears and hunters in the stars, are these patterns real or just happen by chance? It can be proved that if you have enough stars, you will find any pattern that you want to find. In other words, stars are random and they don't have any meaning. So the word then is expected value. The expected value is the number, the estimation of the number of results that you get in a database, assuming that the database was just random letters. The other word that you will find for this is e-value. So instead of looking for a score, in fact, tools like BLAST don't let you choose the score, which is bad from my point of view. But what they ask you is to choose an expected value, an e-value. The smaller expected value, the higher the score. Remember what we say: larger score, give less hit. If the score is larger, you will have less hits. If you have less hits, then the estimated number of hits will be smaller. So we do it from the other point of view. We can calculate, we can have a mathematical formula that will say, look, in this database, in this condition, I expect to have a lot of, I mean, if the sequence are short, if the score is small, we expect to have a lot of hits. If you want to have a hit that is meaningful, you should have an e-value that is small. Typically 10 to minus 5, 10 to minus 20, or even smaller, 10 to minus 60. So if you use something like this, the chances that what you found is random are very small. So we say that we find something that is not random, and maybe, just maybe, it is also biologically meaningful. The big question that you have to solve here, the big question that we have in science is, I will make an experiment, you will make an experiment, and you will get a result. You have three things in that result, three things that are better. It can be the instrument that you are using, the PCR machine, the sequencer, the microscope, something. it can be randomness, noise, let's call it noise, or it can be nature, biologically significant. So what you have to do as a scientist is to make sure that what you are observing is something about nature. It's not something about the instrument, and it's not something about noise. These are the three parts that you will have in every experiment that you do. Noise, signal, and the signal can be decomposed in signal from the instrument and signal from nature. In technology, we care about signal from the instrument. In science, we care about nature. Okay? Therefore,
how do you separate noise from nature, basically?
Well, it depends in every case, but in this case, what we are doing is that noise is something that we can model. Well, under some hypothesis, we can model noise, and we say, okay, if this was noise, you choose a score so high that the probability that you get that score just by noise, just by randomness, is very small.
Okay.
So, for example, okay, let's do an experiment and we will see what happens. But what we do is we choose a small e-value. The e-value is the expected number. This number is always positive. It's either it can be 1 million, it can be 10, it can be 10 to minus 3, minus 5, minus 2. I think 10 to minus 180 is the smallest number that you can have in the computer. There is a limit in the computer, the smallest number, but this number you would like to have it small. If this number is small, if the e-value of the alignment is small, then you know that it is not random. I mean, probably not random. So it's not noise, it's signal. Now, that signal can be biological or can be something else. And that is a different point. These tools do not tell you if this is something in the biology or in the technique, in the instrument. What they tell you is this is not noise. The formula of e-value depends on three things: and the matrix that you're using, the Blossom or PAM matrix, the size of the query that you are using. I mean, larger queries will have a larger score, therefore the e-value will be smaller, and the database size. If the database is bigger, then the e-value will be bigger in general. And the problem is that, I say this point first, the same alignment, if you choose in different database, we have different e-values. For example, remember we have GenBank and RefSec, and RefSec is smaller than GenBank. Let's say you have your query, and your query match a sequence that is in GenBank and in RefSec at the same time. RefSec is part of GenBank. So if you look the sequence in GenBank, your e-value will be larger than if you choose the same sequence in RefSec, even if the result is the same, because it depends not only in the query and in the subject, it also depends on the database size, depending on the context. And then there is another point. Databases like GenBank change every day. Therefore, it is different every day. Some databases change every day. Therefore, the e-value will change from one day to another. Okay? Bottom line, use the smallest relevant database. Of all the databases that are useful for your case, try to use the smallest one. In other words, for example, if we are looking for a 16S database, for a 16S sequence, use a 16A database. If we are looking for a particular protein, look for that protein in the taxonomically relevant group. Okay?